Okay. Hello, Ken Hovind here in Lenox, Alabama, Dinosaur Adventureland. It is December the 13th, 2018. I'm live on the phone with Stefan Frello, and who believes in evolution, and he thinks I do not understand evolution. So tonight's program, we're going to talk about what is the theory of evolution, and the questions that I have, Stefan, from you are, does Kent Hovind really understand evolution and what the theory says? So you wanted to start off. Let's let you go. I'll call up your slides, and yeah, Stefan, tell them who you are, what you do for a living, what your education is, so they know who I'm talking to. Okay, go ahead.
Yeah. Okay, my name, as it says, is Stefan Frello. I'm a teacher at math for 16 to 18-year-old young people who are just about to attend university. I have a master in molecular biology and a PhD in botany. And that is my background. I have debated Hovind for a couple of times already, and I put in the links for our three previous debates, so people can find it if they're interested. And tonight's debate will be on the question we agreed on: does Kent Hovind understand the theory of evolution? And just to make sure that everybody knows what we are talking about, I will start by presenting very shortly what the theory of evolution actually says. It is a theory about descent with inherited modification through natural selection as the most important mechanism, working on random mutations. There are a number of premises for this. First, organisms produce more offspring than will survive. And second, the offspring is genetically variable. The third, some genetic variation influences survivability and reproducibility. And as the fourth premise, new genetic variation is supplied by mutations. And the conclusion based on these four premises is that the genetics of those with better reproducibility will grow in frequency over generations. And that is what inherent modification means. Of course, for this to be interesting, we have to assume, or that is what the theory of biological evolution assumes, that this natural selection will result in accumulation of complexity. And that, of course, is only possible if new genetic variation is supplied all the time, and if some of this new generation is actually beneficial for the organism. But that is the theory that we are working on. The format of our debate, just to clarify, it's not a debate of whether evolution has been proved true or not. It's entirely about what the theory of evolution actually says and whether Kent Hovind understands it. Or rather, Kent Hovind's understanding of it. We agreed on a format where I'll make a short presentation of an example that I think is a misunderstanding on your side, and then we will have a short discussion about that. And when we think that we have exhausted the arguments, we can go on to the next example. We'll try to keep it one topic at a time, so we don't confuse our viewers. Most of my examples are taken from the two videos that I present here. Each of them is about 1.5 hours and can be found on YouTube. Could I go on to the first example, or do you have any comments?
No, go ahead.
Yeah, okay. The first is about how is phylogenetic reconstruction actually made. Phylogenetics is the way various organisms are related to one another. We both agree that at least some species are related to one another. For instance, all the species in the horse family, we agree that those species are related to one another, and at least some of the species in the cat family, some of the species in the dog family, and so on and so forth, are related to one another. The question is rather, how far does this go? According to evolution, this goes as far as it gets. Everything, every single organism in the entire living world is connected by inheritance. Of course, if you take a plant and an animal, you have to go very, very far back in time to find the common ancestor, but eventually it will be there. According to creationism, at least as I understand it, you must correct me if I'm wrong, but according to creationism, we have separate created kinds, and each of those kinds have then, or at least some of them, has resulted in some variation, some different species, but still very, very limited variation, as for instance, the different species in the horse family. But here is your presentation of what the theory of evolution says about phylogenetics. You have these two slides in your presentation, and then it comes. Penicillin only got 2 chromosomes, so they must have evolved first. And a little later, maybe we will have enough chromosomes to be a fern. But this is simply complete nonsense, because chromosome numbers has never ever been used by biologists in phylogenetic reconstructions. So there is nothing in the chromosome number as such that tells you about who have evolved into whom. And there are very good reasons for that. I could go on. Several other examples. Body weight, age, top speed, gestation period, I think it's pronounced. And all of these you use as if evolutionary biology use, or as if biologists use this kind of information for building phylogenies, for reconstructing which species are connected to which. And then you say, they just make up these charts to say how we evolved. But no, they don't. You made up these charts. And none of these suggestions has ever been used by biologists. It's simply just stupid ideas. Because single one-dimensional measurements like these are never used in phylogenetics. It's a bit more complicated than that. So, on to your comments.
Yes, thank you, sir.
In criticizing evolution, I think you would know that phylogenetic reconstruction, how it is done, and it's not done the way you suggested. So please tell us instead of this strawman nonsense, how do you think that scientists actually make phylogenetic reconstructions? Or perhaps you should just admit that you don't know.
All right, my turn?
Yeah.
Okay, Steve, I click on me?
Yep.
There. Okay, thank you, sir. If, Stefan, let me ask you this. If the charts that I made, which were obviously done as a joke, as a humorous example of how evolution doesn't work in those cases, suppose the chromosome number did match what you guys expected. Suppose amoebas were a single chromosome or 2 chromosomes, and humans were the most. If the chromosome number did match and fit your theory, would you use that as evidence for evolution?
No, because
you would not.
Because there are so many different. Well, I can show you examples of very closely related animals that have widely different chromosome numbers. Chromosomes fuse. Whole genomes, at least, well, not so often in animals, but often in plants, whole genomes are duplicated, so that we have one generation of a plant that has perhaps 24 chromosomes, and then the next generation has a double number. And in animals, that is thought to have happened a few times, but much more often chromosomes actually fuse and result in a reduction in chromosome number from one generation to the next. And that has happened quite often, and I can show you some examples if you're interested.
Well,
therefore, the chromosome number as such doesn't tell you anything about the complexity of the organism.
would, instead of chromosome number, if it was the total genetic material, because some chromosomes contain more genes than others, I understand. Suppose we went through all the animals and plants in the world and added up how many genes they have. Obviously, you know full well that also would not match the evolutionary expectation.
No, it wouldn't. It wouldn't, because
genes are very complex.
Genes are lost and genes are gained, and this happens all the time. So it very much depends on whether you gain more genes than you lose, how many genes you have, and of course you have to have quite a lot of genes to form an animal, because an animal is, and even the simplest animals are very complex. So of course you have to have very many genes to form an animal, but exactly how many genes doesn't tell you very much about how complex the animal is, because genes are lost and genes are gained.
Would you agree that with computer codes, the more complex codes would have more lines of code, the more complex programs? Like, does Microsoft PowerPoint probably have more lines of code than, what's a simple program, Steve, some of the early ones?
Calculator.
Calculator. Okay, a calculator on your computer would have less lines of code than, say, a complex program. In this case, it's quite obvious, as well as being intuitive, that the number of lines of code does indicate more complexity. Why wouldn't it, with the more lines of code, which is the genes, why don't the animals and plants follow the expected pattern of more complex is more lines of code? You know it's not that way. Why isn't it?
Well, that is actually, I think that's more or less what is called the C complex question, that the amount of DNA or the amount of genes doesn't exactly match the complexity of the organism. But the problem is, or the answer probably is that the amount of DNA, or the DNA content of an organism behaves in some way independent of the organism as such. There are mechanisms in the DNA that can result in duplication of the larger or smaller parts of the genome, so that you can build up quite a large amount of genetic material without adding any complexity to the organism. So therefore there is no direct connection, and also a very, very large amount of the, as you probably know, the actual genes, the protein coding part of the DNA is only a small fraction. It's only a few percent at most of the entire genome. Most of the genome doesn't form protein coding genes, and a large part of the genome is actually just repetitive sequences that just goes on thousands and thousands of copies of the same short DNA sequence, which probably has no function whatsoever. So,
Stefan, you have a PhD in botany. You know full well some of those, they're constantly finding that those things do have uses. There's no such thing as useless genes that have been proven to be useless. They just didn't know the function of them. It's like back in 1925, they had 180 vestigial organs they said the body doesn't need. And now they found a use for every single one of them. So the fact that somebody says there are extra genes that don't seem to do anything, some of it's simply redundancy for in case of one breaks. But call up your first slide, if you would, please, and let me show you what you said you believe. Call up your first slide and I'll show the people.
This one?
Yes, sir, that's it. Organisms produce more offspring than will survive. That's generally true. The organism is genetically variable. I agree. Some genetic variations influence survivability. I agree. New genetic variation is supplied by mutations. I disagree, of course, but are you saying that when you get different genes, you are more likely to survive? So those with more genetic code would indeed be more complex, but they don't follow the expected pattern based on your evolution theory. You guys believe that genetic variety is how it has to happen. To go from an amoeba to a whale, there has to be a whole lot of new genetic information added. Whales are not amoebas, and amoebas are not whales. Something changed in your theory. Somewhere along the line, a whole lot of information had to be added to change from an amoeba to a whale. So when I make my charts, which are obviously done in jest to show the stupidity of the evolution theory, saying the number of chromosomes, or we could have done the same with the number of lines of gene code, or the number of offspring they produce. Why aren't the more complex animals producing the most offspring? Why do mosquitoes produce so many more offspring than humans? And so I did that knowing full well it does not match your theory. I'm sorry, go ahead.
It was just that most of the the young mosquitoes were eaten by birds before they have a chance to reproduce. So the number of offspring, when you compare different animal species, are not that interesting. It's the number of offspring directly competing to each other within the same species that are interesting. Well, when you look at the evolutionary process, but I have an example from my PhD, because part of my PhD was done on garden plants called crocuses. I hope you know that, I guess. And it turned out that the genus Crocus is very interesting when it comes to its chromosomes, because
hold it. Before we get into that, let me finish answering your question.
Okay.
Your question was, do I understand? And you said I do not understand evolution because of my examples of chromosome number, weight, age, gestation period, how long they're pregnant, things like that. I lined up those various animals in the order that they do appear, like how much do they weigh? Obviously, a whale weighs more than an amoeba, and so I lined them up in the order that they by weight, just to show this doesn't follow the evolutionary pattern, that how long they live, how many offspring they have, the number of chromosomes, the number of genetic lines of genetic code. There are hundreds of ways to line animals up that do not match the evolution. I'm well aware that you guys select, you pick and choose the ones you want to match your theory, but hundreds of other ways you could arrange them don't match, and so you don't use them and say, well, that's not evidence. Well, then that's not.
There's absolutely no reason why any of these suggestions should match what you think that the theory of evolution would say. There's no reason to think that animals during the evolution only should grow larger and larger. Why should they? Why couldn't it be beneficial to be smaller and smaller? Why should it always be better to have a long life than a short, very active life? Breeding within the first very short period and then using all your energy on bringing up your offspring. Why shouldn't that sometimes be better than living a very long time like whales do, and wait several years before you breed for the first time? It simply does not make sense to postulate that evolution should expect life expectancy only to go up and up and up. We don't expect that. There's no reason to expect it. There's no reason to expect chromosome numbers to go up and up. There's no reason to expect top speed to go up and up and up. Sometimes it's better to kick yourself into the ground and stay there.
All right, so you're completely missing my point, I think, deliberately. You are correct that I made up these charts, okay? I made them up intentionally as a spoof on evolution to show that you guys are going to pick and choose. If the evidence seems to support your theory, you will want to spread it and teach it. If it does not support your theory, like my charts do not, then you would say, well, we would not expect it to. Well, this is common sense 101 for a person with a brain that would say, hold it. Yes, those that have evolved the farthest should have the most complexity. They should have the most lines of genetic code. They should have the most chromosomes. Why does a fern have 12 times more chromosomes than a human?
Because
some have 1,200 chromosomes. That's in every cell.
There is no such expectancy. And actually, what you said, the way you put it, actually is very telling, because you said the one that has evolved furthest. No, we all have evolved equally far. We have all organisms living today has exactly the same number of years of evolution behind them.
Would you agree?
Some has accumulated more complexity than others, but we have the same number of years behind us since the first
4.6 billion or whatever number you're using this today. Okay, would you agree that there are some animals that are still alive that appear to be unchanged from millions of years ago? Is the cockroach very similar to the cockroach of millions of years ago? Is the clam very similar?
Yeah.
Well, why didn't they evolve? What happened to them?
They probably have a construction that seemed to be very, very well functioning, and therefore they survived without much change. Actually, that has been a great debate in evolutionary theory, especially, I don't know if you've heard about Stephen Jay Gould, but he has this, he has worked a lot of how come that we see so much stability, and because stability is actually the most widespread thing in biology. Most species tend to stay very stable. So it seems that something has to happen in order for change to happen. The so-called, what should we say, the default is that nothing happens. The default is stabilizing, that most mutations are wiped out by selection, and most genetic change is not changing the anatomy or the complexity very much. But then, once in a while, something happens that speed up this process, and the complexity or change in the anatomy of the animals do take place. But the most of the time, most species stay more or less stable.
Okay, Stefan, hang on, my turn here.
Yeah.
Can't you see how the creationist would look at this exact same evidence and say, as you said, stability is widespread in biology. Almost all plants and animals are doing fine just like they are. There's no need to change. Why can't you see that a creationist would say, yes, they were all designed fine. There's no need to change. They were made fine. They were made complete and perfect at the beginning. The reason the cockroach hasn't changed in 200 million years is because there's no change necessary. Cockroaches are doing fine. To me, what you're giving is clear. It was created perfect.
I can easily understand that a lot of people believe in that, and that was the way biology was until about 150 years ago. Everybody was very confident with that. Species was stable, no change. It took a while before biologists realized that some species have gone extinct. That was a great topic in the beginning of the 18th or 19th century, whether extinction was actually possible, because everything was thought to be so stable. So I can easily understand that, and the evolutionists has no problem with stability. Of course, we see it all over the place. We have still sharks, we have still bacteria, we have still moss and other things that have been with us for a very, very long time. It's not a problem. The problem is how to explain the change, and that is what Darwin did 150 years ago. But back to my actual question, because do you actually know how phylogenetic reconstruction is made? Because
yes.
Even within what you would call created kinds, if there has been speciation, then a phylogenetic reconstruction would be relevant. Even within the horse family, which you repeatedly have accepted as having a common ancestor, the horse and the donkey and the various zebra species must have had some kind of history of speciation behind them, and that history should be able to be reconstructed. So how do we do that? If we just keep it within the horse family, for instance.
Okay. Well, let me ask you a question. Do you believe that the horse came from a single-celled creature like a bacteria?
Yeah.
Are there still single-celled creatures like bacteria alive today?
Yeah.
Why didn't they all turn into horses?
Why should they?
Well, you're the one who said there is a need stable, but there's a need for change once in a while. Something happens causing them to need to change. What made the bacteria change if bacteria are still alive?
Yeah, but there's no, some of them were in a situation where it was a very, very good idea to turn into something else than a bacteria. And actually, well, it's a complicated story, of course, but
I think it's a fairy tale.
The world is, I think you should look at this. You have to look at it in a population kind of thinking. Also, it's easier if we talk about animals evolving into other kinds of animals instead of bacteria, because that's kind of another story. But if you have, for instance, at some point you talk about three-toed animals, which are supposed to be the ancestors of horses, and then evolving into one-toed horses. And what you have to think about in these terms is that we have perhaps a widespread species of three-toed animals. One of the divided into separate populations, which has only limited contact with each other. And one of those populations lived under circumstances where a reduction from three toes to only one toe was beneficial, and that was the population that evolved into modern horses. All the other populations did not live under such circumstances, but lived under other circumstances, so they did not evolve into one-toed horses, but stayed with their three toes. So you have to appreciate that even a single species, a widespread species, live under very, very different circumstances, and some of those circumstances might trigger the evolution from one kind of animal into another kind of animal. But the rest of the species, the rest of the populations of the same species, do not actually evolve at the same time.
I understand completely that that's what you believe. Now, let me point out some things that I think should be obvious. Mm-hmm. Going from three toes to one toed is losing information, less genetic and material code required to make one toe as opposed to three toes. Number two, you're not explaining at all how it got three toes to begin with. Bacteria don't have any toes at all. And you said you believe horses came from something like a bacteria. Somewhere along the line, it had to go from a bacteria to an animal with three toes. And then, so it had to go up to three toes and then lose them back to one to get a horse. Do you understand how all of this looks like a fairy tale, a magic story? It just happened. You have to imagine long ago they faced a problem. There are still three-toed animals today. There,
yeah.
And there are today. I don't think horses.
If some of our audience is very, very interested in the evolutionary history of the vertebrates, which is what you tend to talk about, then they should look into the scientific literature, which has tons of information of fossils found that represent all kinds of different transitional forms between fish and amphibians, and between amphibians and reptiles and mammals, and back in time between fish and something that is more or less like what is now called a lancelet, which is a very primitive kind of not quite vertebrate animal. And yeah, there are so many different intermediate fossils found to illustrate this transition between different kinds of animals. But I actually want to keep getting back to my question: how is phylogenetic reconstruction done? How do we do it? If you have the horse family, for instance, and you want to know how is the different species of zebra related to the donkeys or related to the domesticated horse, how is that done?
You have to understand, all they can possibly find of these creatures is fossils, which would be the bones. They're not finding the lung structure or the circulatory system or the
sometimes they do. Sometimes they actually do.
Okay, that would be extremely rare.
Yeah, very rarely. But sometimes they find of course, not complete structures, but impressions of lungs and other soft tissues, so that you can actually make out at least some detail of how they look.
Well, you're starting off.
Yes, that's right.
You have some very obvious assumptions in your theory. You're assuming that animals change at all when all the evidence seems to be stasis. They seem to be horses always produce horses, cows always produce cows, bacteria still produce bacteria. So you're assuming there needs to be a change to something else. Secondly, you're assuming there's a mechanism that will make them change, and you think added mutations is now what you're saying is what's going to drive this. Well, all observed mutations are harmful, fatal, or neutral.
No.
Nobody's seen a beneficial mutation, and you would need trillions and trillions of beneficial mutations to turn from an amoeba to a horse. And you can't, I don't think you can demonstrate one beneficial mutation.
Well, I would ask people to watch one of our, I think it was in September that I went through the arguments that prove that beneficial mutations are all over the place.
Well, refresh my memory for the audience sake here. What is the best beneficial mutation that you know of?
In Drosophila, that is fruit flies, there is the most well-known fruit fly, or actually one of the most well-known animals at all, is called Drosophila melanogaster, and a lot of these flies has been sequenced. Their complete genome has been sequenced, and other species of Drosophila has been sequenced too. And these flies look so much the same that none of us could tell one species from the other. So I guess that you would accept that they had a common ancestor, and you would point out that common ancestor was a fly, and it was. But in Drosophila melanogaster, some populations has genes that are not found anywhere else in the living world. And it can be shown by very minute genetic investigation that the sequences that make up these genes actually are found in the other species also, but they are not producing any genes there. And if you look very closely to the sequence of these genes, or these of the protein coding gene in one of the populations of Drosophila melanogaster, and find the corresponding sequence in the other species and in the other populations of Drosophila melanogaster, then you can see that these genes are actually new genes. They are not lost in the other populations or in the other species. They have been mutated into existence. They were not there before, but now they are there. New functional protein coding genes with a function. And some of them even has been shown to be very important for the reproduction of these flies. So, and there are many of them, there are hundreds of them. So it turns out that the more detailed you look at the genetics of an animal, the more obvious it becomes that the new genes are popping up all the time. And some of them, not all of them, most of them are probably lost again very fast, but some of them are beneficial, and some of them even turns into being absolutely necessary for the animal. So yes, beneficial mutations have been shown to exist all over the place.
Okay, now maybe you can refresh my memory. I don't recall the details on this. What is the generation time of the Drosophila, the fruit fly? How old are they when they have their babies? How many generations do you get per year? Or
I don't know, 20 or something like that.
Okay, so
I know where you were leading this. Why haven't they turned into something else than a fly? They haven't. Great question. I don't know why they haven't turned into anything else than a fly. So what?
Okay.
So what? And
okay,
I still haven't got an answer to my question. How do we make phylogenetic reconstruction? For instance,
I'm answering your question. You start with the assumption, first of all, that it did happen.
Within the horse family, we agree that it did happen.
No, I don't agree at all. But you start with the
horses are related to each other.
Evolution did happen.
Don't we agree that zebras and horses and donkeys are related?
Well, let's go back to the fruit fly here. Stay with one at a time. Do you believe that, let's say they get a new generation every 2 weeks? I think that would be reasonable. So you get about 25 generations per year. They've been in the laboratory inducing mutations, forcing them to have mutations by changing their environment. You said something happens to the environment to make them need to change, like, colder weather, warmer weather, lightning strikes or whatever, okay? Poisons introduced to the water supply, I don't know. I agree, things happen to the environment that make things have to respond, or some die off. So here they've been in the laboratory for many years, I'm going to guess nearly 100 years, getting about 25 generations per year. So let's say they've gone through 2,500 generations of these fruit flies. They have induced some mutations that you think are beneficial. Are any of those flies, any of the ones with beneficial mutations, more likely to survive in the wild? If you just turned them all loose out of the laboratory, can you name one beneficial mutation that helps it survive in the wild? Is it beneficial to the fly in the laboratory? I don't know, but I'd like to know, where's the scientific evidence, A, that this can happen in nature, and B, that it has happened in nature, and C, that this is going to change a fly into a horse or a whale or anything other than a fly. Stefan, all they've ever gotten are flies, and nearly all of them are dying, or let's say babysit them. They have flies with curled wings, flies with no eyes, flies with no wings. I covered that on my video number 4. All they get are mutated flies, and if you think it's got some beneficial gene someplace, well, yay, but it's got 10 million to 1 negative genes. So it's like saying if we take 1 step forward and we slide back 7 steps, eventually we'll get to the top of the hill. No, you'll never get to the top of the hill. If you take 1 step forward and slide back 7 steps, you're not ever going to get to the top of the hill, and you're never going to turn a bacteria to a whale with this process you're describing, where 1 out of 1,000,000 mutations is beneficial, if that's true. It's not going to get you to the top of the hill, Stefan. You're not going to turn a bacteria to a horse or a whale or anything else. I know you believe it, so you guys make up these family trees based upon A, the assumption that it did happen, and B, the assumption that mutations are going to make it happen. And I'm pointing out there is no scientific evidence that any number of mutations is going to do this. The flies did not gain anything that is not already in the fly category. Flies already have legs and wings. They didn't get feathers. The flies didn't get fins to swim in the water. They're still a fly. Where is the beneficial mutation that's going to change to add some new feature that isn't already in the fly population?
You are sidestepping the question. I am not going to respond to that. I have already explained to you that beneficial mutation has been found. They are found in wild populations. They are not these. This example I mentioned is not from laboratory strains. It's from wild populations. So I'm not going to go any further into that. And I will insist that you answer the question. How is phylogenetic reconstruction made within what you would accept to be a created kind? For instance, the horse family.
Okay, you told us already that organisms have more offspring than will survive. That is, I think, generally true. You told us that genetic variations will survive. Some have genes that allow them to be more adaptable. So the animals are arranged in an completely imaginary order, going from less complex to more complex, from a single-celled amoeba up to a whale. Every night on my YouTube, Kent Hovind Official, I show the chart that the evolutionists themselves make. It's in all the textbooks, showing a family tree from a single cell to multi-cell to plants. They arrange them in order of how they would like them to happen. They are never found in that order, and they are never seen to change into anything else. All the evolutionists do is use imagination. It is SpongeBob 101, in my humble opinion. So yes, you are arranging animals, all of the evolutionists are, in an order of increasing complexity based upon, that's why I point out, why don't you use the number of genes? Why don't you use the number of chromosomes? Why don't you use the gestation period, or how fast they can run, or how much they weigh? Because those things don't fit your theory. So of course you're not going to use that.
Then what do we use?
That's a great question. You tell me.
No, it's not. It is an extraordinary simple question. You can use about half an hour using your Google, and you can come up with a number of scientific papers that tells you exactly how this is done. You've done that, Mr. Hovind. Have you enough interest in this question actually to work out how is phylogenetic reconstruction done? If you don't have done that, then admit that you don't know.
Show me what Google says about phylogenetic reconstruction. What is it that you are driving at? And I will show you the errors in their logic.
But I am asking you within what you would accept as created kind. If we take the horse family, you repeatedly use that as an example of a created kind. Okay? So in your fairy tale story, to use your own words, all living horses, zebras, and donkeys today descend from a single pair of horses on board Noah's Ark. Isn't that correct? That is what you believe?
The Bible says clearly 20 different times the animals will bring forth after their kind.
Could you please answer my question, Mr. Hovind? Don't you, isn't it true that according to your belief, all horses, zebras, and donkeys today, alive today, descend from a single pair of horses on board the ark?
Probably so. There may have been 4 horses on the ark, or 6 horses. I don't know how many kinds. Let's say there are 2. I would agree that 2 horses could produce the horses, the zebras, and the donkeys. Okay. I do not agree that 2 amoeba can produce whales and horses.
No, but that's not my question. My question is, if we then are interested in the actual history of speciation within the horse family, we assume that they started with 1 pair, a male and a female, because that's what the Bible says. 1 pair of horses on board the ark, and then they spread all over the world or whatever, and they start to speciate. And some of them turned into zebras, and some of them turned into donkeys, and some of them turned into what is now the domesticated horse. But there has to be some kind of history. They have to be related to one another in some kind, in one way or the other. How do we reconstruct that history? Just within the horse family.
Well, I think there are some horses that have more ribs than others. There's the shorter horse. I think it's Mongolia, if I recall. They have 1 less pair of ribs, or 2 less pairs of ribs. I think you could easily... Pardon me?
So what?
Well, since your PhD is in botany, maybe we should do something more in your field. But
no. Stay at the horse family, Mr. Hovind. Now answer my question. Do you know? Yes or no? Do you know how we could reconstruct the phylogenetic history of the horse family? Yes or no?
Reconstructing the history of any creature, of course, would require a lot of guesswork and a lot of imagination. It's like reconstructing the history based on...
simple way to do it. My whole point is that you don't know.
I think I do. I'm trying to explain.
You claim that you understand evolution, and you don't know the very, very basic thing. How is phylogenetic reconstruction done? It's a very, very simple question that can be answered with 1 single word. And you
Imagination. Okay, my word is imagination. That's how it's done.
No, it's not.
Okay. They imagine that the whale came from a bacteria.
I'm not talking about whales and bacteria. I'm talking about horses.
Okay. You imagine that a horse came from a bacteria over billions of years.
Hovind, stay at the topic. Don't.
I'm on topic.
No, you're not. I'm talking about
you know it, and that's what bothers you.
Horse family.
I'm showing the stupidity of the idea that you think a horse could come from a bacteria. I think two horses on Noah's Ark could produce the horses, the zebras, the quagga, the mules, crossbreeds, the shorter horses, the longer horses, the taller horses, the smaller horses. I think all the horses could easily be put into a family tree, a phylogeny, and say, yes, these are probably all related. You could do the same with dogs or apples or anything else. But you guys then take that and go wild with your imagination and think, aha, see, this proves horses came from a bacteria. This is where you jump from the science to imagination, to a religion. You believe it, and I feel sorry for you for having been taught such a thing, and you actually had to pay to be taught that stuff. So, yes, you do believe horses came from bacteria.
Mr. Hovind, you are so much stressing my whole point. You don't know. You simply don't know how.
Stefan, I told you, I do know clearly. It is imagination.
So
that's how you guys do it. You imagine that a horse.
So if we want to reconstruct the phylogenetic history of the horse family, just the horse family, we agree that all horses come from a common ancestor. And now we just want to reconstruct the phylogenetic reconstruction, the history of the horse family. No whales, no bacteria, just horses. Do you know how this would be done?
I think they would look at the variety of horses from a variety of sides. Let's look at the circulatory system. Let's look at the respiratory system. Let's look at the reproductive system. Let's look at
all that would be exactly the same.
All those would be very similar between a horse and a zebra. The integumentary system, obviously a little different. The zebras have stripes, not many horses do. I think you could look at the number of chromosomes and say, wow, they're pretty similar. You could look at horses and mules have eyes on the outside of their head instead of straight forward. A binocular vision, they have, I forget, peripheral vision, I forget the name for that one. Their eyes are on the side of their head. I think we could arrange animals like that and come to some conclusions that, wow, these are the same kind of animal. The horse, the zebra, the short horse, the Mongolian horse, the Shetland pony, the Clydesdale could have easily had a common ancestor. And that's where that is correct. We could do that with buildings or with tools, you know, in the shop, or there's all kinds of ways to create a phylogeny, a family tree. I could create I think I could probably what tools came from what factory.
Do I have my slide on the screen now?
This one.
The slide with the chromosome number of the zebra and the donkey.
Yes,
and so on.
I have it up on screen. Go ahead.
Yeah. Okay. So these are the chromosome numbers of the different species within the horse family. As you see, they are widely different. And just looking at the animals, you would probably expect that the three species of zebra was more closely related to each other than to any of the other species. But as you can see, the chromosome number doesn't tell you anything about that kind of relation. And what we actually do, Mr. Hovind, now that you have so clearly demonstrated that you have no idea how phylogenetic analysis is actually done, then let me tell you. We use DNA sequences, Mr. Hovind. And the rest of you, here is phylogenetic. I have the reference in the bottom of the slide, based on mitochondrial DNA and some nucleotides, and the lines to the left show the relationship. The plains zebra, the imperial zebra are closer related to each other than to anything else. Next is the mountain zebra, and then the Kiang and the Kulang, they are donkey-like animals, are closely related and a little less closer related to the donkey, the domesticated donkey. And we have the domesticated horse and its wild close relative, Przewalski's horse. And this is the way it is done. DNA sequences, not chromosome numbers, not gene numbers, not anything else. DNA sequences. And the reason that it is a very, very reasonable way to do it is that what is actually inherited from one generation to the next is exactly DNA. So the variation that you see between the zebras and the donkeys and so on is based on the variation in DNA. So why not go to this to the corner of the question that if you can look at how the DNA, how the DNA is related, how the level of identity between the DNA from different species. If you look at that, then you have a very, very good measure of the actual phylogenetic history of this.
Okay, now, Stefan, let me ask you a question. Would you agree? Would you agree that back in Darwin's time, 1850s and 60s, the microscopes were not sufficient to see much more than a cell? Darwin thought a cell was just simply a little glob of jelly. They did not understand. Nobody knew what DNA was. When was this mitochondrial DNA discovered at all? And when did they start doing these sequences?
Until about 40 years or so, 30, 40 years or so, it was phylogenetics was done with a very, very detailed anatomical description. They looked as many details of the anatomy as they in any way could come over. But I'm pretty sure that until DNA was used for phylogenetic analysis, it was very, very difficult to come up with any good idea of how the zebra and the donkey and the horse was related, because they are so close.
Right.
They are so much identical.
I don't want to put words in your mouth. I want to try to explain to the audience what you just said. For most of human history, since Darwin's, anyways, for the first 100 years after Darwin, they arranged animals based upon anatomy.
Yeah.
And now they just started 40 years ago arranging them based upon mitochondrial DNA sequencing.
And other things. And the interesting story is that they came up with almost exactly the same history as they have built on anatomy. That's very interesting.
Well, that's your belief, I'm sure. That's why in my charts I pointed out, which were obviously a spoof about why didn't they use gestation period? Why would you use anatomical similarities to prove common ancestor? Why not use anatomical similarities and DNA similarities to prove common designer? Stefan, would you agree that these similarities in DNA, mitochondrial DNA, could prove the same designer wrote the code? Is that even an option in your thinking?
Yes, of course.
So it could be the creation, which says they're going to bring forth after their kind, is supported by the similarity of mitochondrial DNA, just like it is supported by the similarity of anatomy.
No,
they
it's
used
not.
for 100 years, and now they use DNA. I don't know what they're going to use next week, but all those things could just as easily be interpreted as evidence for creation.
No, it couldn't, because if you, for instance, look at the horse family is part of the octoed ungulates. There are three families of octoed ungulates: the tapirs, the rhinos, and the horses. And if you look at the mitochondrial DNA from tapirs, rhinos, and horses, you will see that they match each other at a higher level than they match any other animal. Now, why on earth should the mitochondrial DNA, which has absolutely nothing to do with anatomy, it is very, very basic biochemistry, it takes care of. Why would mitochondrial DNA from a rhino look more like that from a tapir or a horse than any other animal? And coincidentally, that is exactly the octoed ungulate order, which has been recognized, I think, by Linnaeus 250 years ago. Isn't that odd? Isn't that just very, very strange from a creationist point of view, that the mitochondrial DNA looks exactly as it should if evolution was true?
It looks exactly as it should if creation is true also.
Why? Hey, stop there. Why? Why should you, as a creationist, expect the mitochondrial DNA from a horse to be closer more identical to mitochondrial DNA from a rhino than it would be to the mitochondrial DNA from a cow. Why?
Yeah, our hour is up, but let me try to answer that question for you. The similarity, Microsoft Corporation, I don't know, Steve, how many different programs have they produced? Microsoft PowerPoint, Microsoft Word?
Sidestepping the game.
Thousands and thousands, okay. Microsoft has produced thousands of programs. Some of them just games, right? Some of them high-tech military stuff, I'm sure, okay? How many of those have similar lines of code? How many have identical lines of code? Probably. Now, does the size of the computer that it fits in, can we determine anything from the anatomy of the computer, what's in it in the code? Or can you store that same code in a variety? Can I store it in a phone? Or can I store that same code in a computer the size of a house? Or a computer the size of a suitcase?
You're completely sidestepping the question.
No, I know I'm not. I'm trying to illustrate, Stefan, so you can see it. You're trying to say we arranged them by anatomy. Well, we could do that with computers. We could arrange the smaller cell phone, which is a computer, up to the giant, Cray computer. We could develop a phylogenetic tree of computers, and we could also develop a phylogenetic tree of the amount of code it will hold. And we try to come up with imagination. See, all of that is imagination. You're assuming the animals changed at all. We never see it happening today. No horse produces anything but a horse. No bacteria produces anything but a bacteria. There are no examples of it happening. It only happens long ago and far away. This is your religion. I wish you could see that.
You don't even address my questions. You haven't. I waited half an hour for you to just come up with one simple word. DNA sequence. Mr. Hovind, let me finish this.
No, it's imagination.
I waited half an hour for you to come up with one single answer to my question.
I gave you the word.
Was DNA sequences, and you didn't come up with it because you don't know. And now I'm asking you, why would the mitochondrial DNA from a horse be more similar than the mitochondrial DNA from a rhino than it is from the mitochondrial DNA from a cow? And you don't answer the question. You start talking about something completely different. Why are there any pattern in the level of identity between these mitochondrial genomes? Why is it not just a complete mess? Why are there such a very, very fine-tuned pattern that fits exactly what you should expect from evolution?
The horse and the rhino have some of the same needs. They both need to walk on 4 feet. They both need to have a balance system. They both need to see. They both need to grow hair. So of course there are similarities in the DNA. They have lots of.
So does the cow. That's not an answer to why the mitochondrial DNA from a horse should be closer to the rhino than to the horse.
Well, if you want to believe that evidence you're presenting is evidence that we all have so you believe a horse and a rhino came from a bacteria. Would you agree that the DNA mitochondrial sequence of a bacteria is different than a horse?
You are sidestepping the question all the time. You are simply not addressing the questions that I asked you.
I'm pointing out the stupidity of it. I think my audience is
not. No, you are not. You are proving that you don't know what you are talking about because you have no answer.
I gave you an answer. The answer is
you didn't even address the question.
Stefan, the single word answer I would give is imagination. Imagine. You guys think a horse came from a bacteria, and you're looking desperately for evidence to support that. And when you looked at anatomy, oh, you found what you're looking for. And when you looked at DNA sequencing, oh, you found what you're looking for. But if you look at gestation period, or look at body weight, or look at lifespan, or look at number of offspring that's in my chart, well, that doesn't fit. So therefore, that's not what we're looking for. So we won't accept that. It's imagination. Hey, let's continue this another time. We've gone over an hour. I got folks waiting on me here. So let's go over this another time. You asked a question, do I understand how you do the sequencing? And my answer was imagination.
So DNA sequencing.
Pick another question. We'll do it next time.
Okay. Am I allowed to finish this?
Yeah. Go ahead and give a closing statement. I got folks waiting here. Go ahead.
Okay. I think that if you by now turn back an hour and listen to our conversation once more, you will find out that Hovind didn't answer one single of my very, very simple questions. which he would have been able to do if he had paid just a little bit of interest into what evolutionary theory actually says. But Mr. Hovind has no interest in knowing what evolutionary theory or phylogenetic analysis actually is, because if he knew, he knows that he would have to lie to people in order to maintain his stupid stories about carrots going into chickens going into ferns or whatever, which has absolutely nothing to do with how evolutionary theory works or how phylogenetic analysis works. It was crystal clear. I waited about half an hour for an answer for how phylogenetic analysis was done, and there was no answer because Kent Hovind doesn't know. I asked the question several times, why is a zebra or a horse closer related to, or genetically closer to a rhino than to a cow, and we got no answer. So I really hope that people will listen to this once more and see how you simply sidestep the questions all the time. Thank you.
Well, thank you, Stefan, for coming on. Let's do this again another time. I think I did answer your question. I think evolutionists for 150 years have wanted desperately for their theory to be true because they want to get rid of God. The Bible clearly says the animals will bring forth after their kind, and that's what the evidence has shown for all of human history. Every farmer on planet Earth will point out that it works. Whether it's corn, you plant corn, you get corn, you cross cows, you get cows. There are no exceptions. The word that describes evolution, the best of any word I know of in the world, is imagination. You imagine that horses and zebras and rhinos and cows and bacteria and fish and birds are related and all came from a bacteria. That is a religion in every sense of the word, in my humble, totally, completely unbiased and correct opinion. So thank you for joining us, folks. Stefan, let's call the secretary and schedule it again. I'm sorry, my schedule is just really hectic here. We got a lot going on, but come on over and visit Dinosaur Adventureland. We'll put you live on our program, and you can sit beside me. We'll have the camera on, and you can explain why you think horses and rhinos came from bacteria. I would love to hear about that. I'd like to see how you demonstrate the genetic mutations that are going to add enough information to change over, I'll give you trillions of years. We'll make it quadrillions of years to get a bacteria to turn to a horse. So keep up, keep studying. Eventually you can be an independent temperamental Baptist like me and give your heart to the Lord. That's what I'm trying, Stefan. I'm trying to win you over to the Lord. Thanks for joining us, folks. We got to go here. We'll see you next time. Stefan, let's do it again. Thank you.